Hello and welcome back. Today we are going to talk about a really amazing aspect of virology: reverse transcription and integration. And this story begins way back in 1908 when the first tumor viruses were discovered. 1908, chicken leukemia virus, and in 1911, Rous sarcoma virus. So the first viruses shown to be able to cause a cancer in these cases, both in chickens. And we'll come back to that story in a couple of lectures. It's really interesting. Rous, in fact, working here at the Rockefeller Medical Institute, discovered this. These were called tumor viruses because they called tumors. Remember, by 1908, we already know what a virus is, pretty much, so they called these tumor viruses. And later on, in the 50s, when we began studying viral genomes, we found that these had RNA genomes, so they were called RNA tumor viruses. So RNA tumor viruses. And then finally, today, we're going to give them another name. Many years later, Howard Temin, a professor at the University of Wisconsin-Madison, had very great insight, which led to the discovery of how these viruses work. He noted, first of all, these viruses cause permanent changes in the cell. You'll see again in the oncogenesis lecture, when you infect cells in culture with these RNA tumor viruses, there is a permanent alteration of their shape and properties called transformation. So permanent changes in cells. And he said, this is an RNA virus. It can't be that RNA does this. RNA can't make permanent changes. He said it must be DNA. So he hypothesized that somehow this RNA got converted to DNA and it became a permanent part of the cell. And that was what he was called his Provirus hypothesis. And as you will see today, we use that name now to mean a DNA copy of the RNA tumor virus genome integrated into the cell genome. And that got him looking for the enzyme that would convert the viral RNA to DNA. So that was Temin. Then on the other hand, we have David Baltimore from the Baltimore scheme. He was thinking about RNA in terms of polarity and whether there's a polymerase in the particle, remember? So he found first that the plus strand RNA viruses, there's no RNA polymerase in the particle because these RNA can be translated as soon as they get in the cell. However, he saw that there were viruses with negative strand genomes, and he reasoned there must be a polymerase in them. And he found that in VSV initially. And then when we started thinking about RNA viruses permanently changing cells and having to make a DNA copy of their genome, he said this must be in the particle because cells don't do this. Cells don't copy RNA to DNA. He was wrong on that side because we now know that cells have an enzyme to do that, but at this time we didn't realize it. So both of them looked for this enzyme. They both published it in Nature at the same time. Here, RNA-dependent DNA polymerase and virions of Rous sarcoma virus by Howard Temin. And you can read this abstract and you'll see the Provirus, the DNA Provirus hypothesis. He thought the RNA had to go to DNA, and he found the enzyme in the particles. And Baltimore, on the other hand, also found it using a different RNA tumor viruses. So they were both published at the same time, and they both got the Nobel Prize just a couple of years later. And I interviewed David for TWIV 100 years ago. Very interesting if you want to get more insight into that. So the enzyme that is in the particle that converts the RNA to DNA, of course, is called reverse transcriptase. And it got their name because they reverse the flow of genetic information. So we thought for the most part DNA goes to RNA and then, of course, to protein. These go from RNA to DNA, which people thought could happen, but they'd never been discovered. And so when this enzyme was discovered, it was called reverse transcriptase for that reason. This was completely revolutionized molecular biology. Every coronavirus diagnosis you read about today is done using reverse transcriptase. It's done by RT-PCR. The RT stands for reverse transcriptase. You take a little bit of fluid from the patient, you extract the RNA, and you convert it to DNA first with RT, and then you amplify it by PCR. It's just amazing. So cloning genes, expressing genes, biotechnology, clinical medicine, research, everything has been completely revolutionized by the discovery of this enzyme. And they didn't patent it when they discovered it. Baltimore and Temin did not patent it, although he did say to me in another interview he wished he had. But it's good. This way you can use it freely, and here is a company that you can just buy it. Obviously, companies make all modified forms that are very efficient and so forth. Really a remarkable enzyme. And today I want to explore the role of this enzyme in the reproductive cycle of not just retroviruses, but also hepatitis B viruses have this in their reproduction cycle as well. So the viruses with RT are the retroviruses that have a plus RNA in their genome, and the hepatitis B viruses, which have DNA in their particles. Retrovirus has RNA in their particles, the hep B have DNA, but they both have reverse transcriptase in the reproduction cycles. So here is a picture of a retrovirus. They look slightly different. This is Rous sarcoma virus, and if I showed you HIV, it would look slightly different. We'll see that later. There on the left is an electron micrograph. It's got a membrane, and by the way, it looks spherical, right? Even though it's not icosahedral. That's one of the questions about half of you got. It doesn't have to be icosahedral to look spherical. It's got a dense core, and on the right is a schematic. So we have an envelope, we have glycoproteins in the envelope, and notice they're sparse. They're not packed together as we see in a lot of the other envelope viruses. And as we'll see later, that makes it hard for antibodies to cross-link them and neutralize infectivity. We have, so those are glycoproteins in there, and then inside we have a capsid. So it's a nucleocapsid because it's within the envelope. And inside there's the genome, which is 2 strands of plus RNA, not 1, but 2, and they're coated with a protein called nucleocapsid. So even though this is a plus strand RNA virus, it's one of the exceptions that's coated in protein. Here's the genome of the virus. Let's look in the middle, that green molecule. That's the RNA that's in the particle. 2 of those, they're capped and polyadenylated. They're messenger RNAs, and they encode 3 general regions: GAG, POL, envelope. The GAG encodes structural proteins like the capsid and nucleocapsid. The POL encodes the reverse transcriptase and other enzymes that are needed for that process we'll talk about. And the envelope encodes the glycoprotein. And for that to be produced, the genome has to be spliced. So a spliced product of the genome gives you the envelope, whereas the GAG and POL are made either as GAG alone or a GAG-POL fusion protein. So these are what we call retroviruses with simple genomes. They don't encode a lot of proteins. HIV, on the other hand, is a lentivirus. It's a subpart of the retrovirus family. Is a complex genome, encodes a lot more proteins, as we'll see. On the top is the Provirus. Now, the Provirus is a word I will use specifically to mean the integrated DNA into the chromosome. That's the only way I will use that. And so now what we're looking at is a double-stranded DNA copy of the RNA. It has on either side 2 LTRs, long terminal repeats we will explore, and in the middle are the GAG-POL envelope coding regions. And you can see how the proteins are encoded on that. So here's a reproduction cycle of these viruses. We start by attaching, of course, to cell receptors. Most of these viruses encode at the plasma membrane, and the nucleocapsid is released into the cytosol, and the RNA never gets out of the nucleocapsid. So it's never translated for that reason. Ribosomes can't access it. If you extracted the RNA out of the particle and put it into cells by transfection, it would be translated, and it would start an infectious cycle because all the proteins would be made. But in the reproductive cycle, that doesn't happen. The nucleocapsid remains intact, and then reverse transcription is completed in the cytosol. You make double-stranded DNA, and then that goes in the nucleus. It integrates into the chromosomal DNA of the host, so now we have a provirus, and it is now transcribed in situ in the chromosome to make mRNAs, including full-length mRNAs that will eventually get packaged into virus particles, and spliced RNAs that give rise to the envelope, as well as some full length that go out and are translated to make the gag and the pole. And we'll come back to the later parts of the cycle next time when we talk about assembly. So let's talk about reverse transcription. This is a very interesting process. I happened to be a postdoc with Baltimore a couple of years after he got his Nobel Prize, and his lab was furiously working to figure out how reverse transcription works. Not only do I have a fond place in my heart for you, but I have a fond place for reverse transcription as well, because I saw it being discovered. So reverse transcriptase, the enzyme, is a primer-dependent enzyme. The primer can be DNA or RNA. The template can be RNA or DNA. You'll see why in a moment. But it only incorporates dNTPs, not NTPs, into the product. It only makes DNA. It does not make RNA. So there's a template and a primer. We've seen that before. Primer is extended in a 5' to 3' direction. Really key point here. Reverse transcriptase is not unique to retroviruses. Baltimore thought that it was, and so did Temin. When they discovered it, it had never been discovered before, but it turns out that you can find reverse transcriptase in bacteria and archaea, as well as eukaryotes, not just viruses, but the cells themselves. And so, as you know, bacteria, archaea, and eukaryotes, the 3 domains of life, have a common ancestor, as shown on this tree. So this reverse transcriptase, by virtue of the fact that it's encoded in the genomes of all 3, must have been in the common ancestor. It was there before the common ancestor split into the 3 domains. We also think that reverse transcriptase was the bridge between the RNA world and the DNA world. Remember, at the very beginning of this course, I talked about an RNA world where RNA molecules only existed. In the very beginning, there were no proteins even. There were catalytic, self-replicating RNAs. And at some point, RT emerged, proteins emerged, of course, and then RT made DNA from RNA, and that started the DNA world. Now, that's a theory, of course, very hard to prove. So, and in terms of viruses, not only retroviruses have RT, but hepatitis B viruses and a family of plant viruses called the Polymerases. And if it sounds like cauliflower, yes, they do infect cauliflowers. And they have reverse transcriptase in their reproduction cycle. Now, there are lots of reverse transcriptases that have been discovered, and they're kind of arranged in this radial diagram here on the right, which is actually a radial phylogenetic tree, and they're colored according to where you find them. There are virus and virus-like reverse transcriptases. Those are the bluish colors. So the hepadna, retro, and polymerases. But then there are also mobile elements in cells that have RTs that are virus-like, like gypsy and copia, found in fruit flies and other organisms. Then we have reverse transcriptases in bacteria and archaea. And then we have reverse transcriptases in our cells, eukaryotic cells. And there are telomerase reverse transcriptases. So remember the ends of your chromosomes, telomerases, have an essential reverse transcriptase in them that keeps the ends of a certain length. And that wasn't discovered until after RT and viruses. And then there are a series of elements in eukaryotic genomes called lines that we will talk about a little bit today that also encode reverse transcriptase. So much broader than Baltimore or Temin ever remembered. We know the structures of many reverse transcriptases, and they were included in this diagram I showed you some time ago. There are 4 different kinds of nucleic acid polymerases, and there we have the RNA-dependent DNA polymerase, the second one from the top, reverse transcriptase. You can see it has some of the same conserved elements in colors as the other polymerases. Again, they all evolved from a common ancestor. And remember, in the catalytic site, the palm domain, this yellow, these 2 beta strands that have the aspartate residues that coordinate the magnesiums. The last question on the exam, right? If you change one of them, the magnesium doesn't bind. Most of you got magnesium, but if you didn't mention magnesium, you lost some credit. They're in the reverse transcriptases as well. Here you go. We have an asp, and reverse transcriptase. Same idea, holding the metal ions for catalysis. All right, so that's the enzyme. We're going to see how that works in a moment. There's a second activity of the RT protein. It's not a separate protein. It's the same protein called RNAse H. And H comes from hybrid because this RNase chews up the RNA when it's in a duplex form. It can be RNA-RNA or RNA-DNA, hybrid, if you will. And it makes endonucleolytic cleavages, which means it cleaves internally, not at the ends. That would be exonuclease, right? And it makes short oligos with 5' phosphates and 3' hydroxyls. So there on the top is a RNA-DNA hybrid, and the phosphate joining two adjacent bases there. And the red arrows are where RNase H would cut. And you end up with a short oligo shown at the bottom here with a phosphate at the 5' end and a hydroxyl at the 3' end. You'll see why this is needed for reverse transcriptase in a moment. Reverse transcriptases are enzymes that look like a right hand. They have a palm and a thumb and a fingers domain. This is the RT of HIV-1, which happens to be a dimer, has two subunits, P66 and P51, that form a dimer in the middle there. And there you can see the palm and fingers and thumb domains. Here's the active site right up at the top left in the palm domain. And this is a DNA-RNA hybrid. So the RNA has come in here and the DNA is copied. And now we have a duplex molecule. And here's the RNase H domain in orange there. So remember, again, the RNase H is an integral part of the enzyme. Here it's part of the P66 subunit. This is a cartoon of the reverse transcriptase that makes it a little clearer. We have the catalytic domain in the palm there labeled, and the two aspartates that coordinate the metal. Here it's abbreviated Me, standing for metal. It can be magnesium, it's physiologically used, but you can add manganese and it will work as well. So the RNA comes in here at the left between the fingers and thumb. A DNA copy is made, and then it passes by the RNase H, which has its own active site, which also depends on two metals for its activity, and it removes the RNA, and out comes a single strand of DNA. So you put a single strand of RNA in, and you make a hybrid for a short time. It exists only in the enzyme, and then as it comes out, the RNA is degraded. This process is very slow, about 4 hours to make the whole genome of the retrovirus, which is about 9,000 bases long. 4 hours, okay? It's a long time, and it makes a lot of mistakes. One misincorporation for every 10,000 to 1 million bases. So essentially, you can have a mistake every time the genome is replicated. And that, of course, has many implications, which we'll talk about when we talk about antiviral therapy, for example. That's why we need to use 3 antiretroviral drugs to hold HIV replication, because if you use one, within weeks you have resistance. First question, reverse transcriptase has revolutionized molecular biology. Which statement about the enzyme is not correct? It's unique to retroviruses, it's packaged in the particle. The RT also has RNase H. The name comes from reversing the flow of genetic information, and it might have bridged the ancient RNA and DNA world. All right, how did we do here? Most of you got A's. What's wrong, it is not unique to retroviruses, of course. It is found in many other organisms. So the question was, what's wrong? So it has RT activity, and the name comes from reversing the flow of the genetic information. Let's talk about the process of reverse transcription. This is very interesting, and you need to know this. Understand it at least. You can always draw it on your sheets for the exam, but you need to understand how it works. So here, there's the particle. There are 2 copies of the plus strand RNA in the particle, and they're linked together by base pairing here on the RNA. So on the upper left in panel A, we have the 2 RNAs by themselves. The 5' ends with the cap are shown there. They interact with each other via what's called a kissing loop complex. So that's a sequence here in stem loop 1. So here on the bottom is the very 5' end of the genome of the RNA, just a few hundred bases. Forms a lot of secondary structure. This kissing loop has a sequence that base pairs with a similar sequence in a second copy of the RNA. So they base pair, and that's what brings them into a particle together during assembly. The other thing that's important here is that there's a tRNA bound to both RNAs very near the 5' end of the RNA. See it right there. It's base pairing with a red sequence, which is called PBS, primer binding site, and that's complementary to the tRNA. You're going to see that that's the primer that starts reverse transcription. So this explains how the two RNAs are brought into the particle. They're base paired with each other, and then they're base paired with tRNAs. So the particle has not only 2 viral RNAs, but tRNAs as well. Why do we have two copies of the genome? We think it's to make the virus genetically resilient. Retroviruses are pretty resistant to UV and ionizing radiation, which typically causes mutations in nucleic acids. And we think having 2 copies allows you to make a DNA in the end that's correct. Imagine here there are 2 copies of our RNA in green on the bottom here. And these are 3 genes, A, B, and C, and the A gene, say, is mutated. What happens during reverse transcriptase is that the enzyme flicks back and forth between the 2 RNAs very quickly. And it makes a copy of a little bit of one, and then copies a little bit of the other. So you can imagine if the genomes have mutations, eventually you'll make one that's free of mutations by copying the other strand. That's a random process, of course. It's not planned in any way. But this is why we think there are 2 copies in the particles, so that the virus can be more resilient to mutation. Well, in the end, of course, they still sustain a lot of mutations, but I guess it would be worse without 2 copies, yes? Yeah, we do call them diploid, yeah. Two copies of the genome, right. All right, here's how the primer is bound in a little more detail. So there again at the top, the 2 RNAs. And we're expanding just the very 5' end with a cap. And there's the primer binding site with the tRNA bound. There, this is shown as a line, but it's not really a line, of course. The RNA is all folded up. Here is the RNA structure in this region without the tRNA. Here's the tRNA, which itself is extensively self-base paired, of course. And then on the right is the tRNA base pairing on the viral RNA. So you can see it insinuates itself into the structure. There's a part of the tRNA that's not base paired. And then the key there, of course, is the 3' end, which is going to be the place where the RT begins to add bases. All right, so that's the tRNA primer. So let's go through these steps and talk about how this works. So this first key point, which I've already mentioned, is that DNA synthesis is entirely cytoplasmic. The virus binds and uncoats at the surface. The nucleocapsid gets in. It becomes porous enough to allow dNTPs to get in the particle. Yeah, but not so that the RNA can get out. And the reverse transcription occurs in the cytosol. You get a double-stranded DNA copy, which then will go into the nucleus. Now, here's the first step. We're taking the RNA out of the particle and simplifying it. We have a tRNA primer bound, and this is weird because it's right near the 5' end of the RNA, which is not where I would put it if I designed this, which goes to show that logic doesn't always happen. But there is a reason why this is occurring. Okay, the tRNA starts as a primer for RT. RT makes DNA. So in these diagrams, the RNA is green, the DNA is blue. And so the RT begins to make DNA, copying the viral RNA, using the tRNA as a primer. And of course, immediately runs out of space. This is only about 100 bases or so from the 5' end. That's called strong stop DNA, because when people were first doing these reactions in vitro, they found a 100 base band on the gel, and they're like, what is this? It's 100 bases long. How could this be of any significance? You'll see. As the DNA is made, the RNAse H is chopping up the template. That's what the broken RNA means there. And then the polymerization continues on the 3' end. So if you look at these sequences, and these are important for you to get a hold of, these little R and U5 Those are sequences at the very 5' end of the RNA. We'll come back to them. Then on the other, at the 3' end, there's also an R, and then there's a U3, which is different from U5. Now, the little r means they're the same polarity. A big R would be complementary. So here, for example, when we make DNA, we're making a capital R and a capital U5. It's the complementary strand. That capital U5R in the DNA can now base pair with the 3' end of the genome. That little r there is complementary to the big R. So these RNAs have repeats at their ends. So that base pairs, that DNA, strong stop base pairs with the 3' end, and it begins to prime there, which is shown on the next slide. So here, we're drawing it as a kind of stretching, but in fact, this is all in a compact structure, so you don't need to do any of that. It's for us, for our graphic view. So now we have RR annealing, and the RT will continue to make the DNA. And as it makes it, it chops up the RNA, which is shown in the middle and on the right. But it does leave behind a piece of RNA called PPT. That stands for polypurine tract, and that's going to be a primer for the other strand of DNA synthesis, which actually starts even before the first strand is done. Right? So the RNAse H doesn't chew it up. It remains behind as a primer for the second strand of DNA. So at this point, we have almost the first strand of DNA, not quite. tRNA is still there. We're starting to make the second strand of DNA, which is in a different blue color, showing that it's the second strand. All right, we continue that on the left. Now we've made the first strand completely. We have copied the primer binding site. As you can see there, we've displaced the tRNA, and now the first strand of DNA is complete, and now the second strand is elongating. It gets to the tRNA. The RNAse H cuts off the tRNA primer at this point. And now that little piece of DNA that has been made is going to be priming on the original first strand that was made. So that's the primer binding site, which is part of the first strand of DNA. And eventually this second strand will copy the entire first strand of DNA. So that brings us to the top again. There's the molecule we just left off. Complete first strand, second strand being made. That second strand will go all the way around. It will make a complete double-stranded copy, as you can see here. And what you end up is the molecule at the bottom, which is a complete copy of the RNA, but it has something that the RNA doesn't have. It has 2 LTRs at each end. We're going to look at the RNA in a moment and compare it. The RNA didn't have a complete LTR at each end. An LTR is U3RU5. U3RU5. That is an LTR. The RNA only has 2 of those elements. So this whole process, this weird process of jumping off one end and the other, it's designed to reconstruct 2 LTRs, because as you'll see, when the genome RNA is transcribed from the provirus, you lose parts of both LTRs. So the sequences that are repeated in the RNA are built there so that the LTRs are regenerated during reverse transcription. Here's the key slide. So here's our RNA that we started with. 5' end, little r, little U5. There's no 3. There's no 3 sequence. So there's no complete LTR at the 5' end. The 3' end R, U3, there's no complete LTR there. The process of reverse transcription generates them. So you go U5, R, U3, that makes 1 LTR when you do the jump from one strand to the other. And so from this RNA, you make a double-stranded DNA, which has an LTR at both ends, U3RU5, U3RU5. In addition, the primer binding site is maintained because it's copied as part of reverse transcription. So there's a plus and a minus strand DNA, 2 LTRs. We'll see what those LTRs do in a moment, but they're really important. So I've made an animation of this, which you can look at. I would recommend looking at it. It's about 5 minutes long. That goes through this in an animated way all at once, and you can see how this process is working. All right, next question. Which of the following steps occur during reverse transcription of retroviral genomic RNA? Priming of minus DNA synthesis by tRNA, 2 template exchanges, degradation of the viral RNA by RNAse H, generation of 2 LTRs, all of the above. Okay, what did we do here? All of the above is right. Most of you got that. Everything is right. Priming of minus, 2 template exchanges, one in the beginning, one at the end. RNase H degradation of the template, and we make 2 LTRs. So let's summarize that again. We started with a single mRNA without any LTRs at either end. We go through reverse transcription with 2 template jumps that generates a double-stranded DNA with 2 LTRs. So that's shown at the top there. And the next step is this is going to integrate into the host DNA. This is an obligate step for retroviruses. So here's the DNA at the top. It's unintegrated. It's going to integrate into a host target, which is colored purple here. That's the host DNA. And then you end up with a provirus, which is integrated DNA copy of the viral RNA genome. 2 LTRs on either side, GAG, Pol, ONV genes, PBS, PPT serves as primers. There are 2 characteristics of integration that you should remember because they are really useful to understand the process. First of all, when integration occurs in the mechanism I'm going to show you in a moment, the host target sequence is duplicated on either side of the provirus. And you can see that by this purple sequence has now become duplicated on either side, shown by the red. You'll see why that happens in a moment when we go through integration. So duplication of the target sequence. The second is you lose some bases at both ends of the LTR. It can be a couple or a few more depending on the retrovirus. Here we're losing 2. We have AATG at the very tips of the 5' or the left-hand LTR, and you can see only TG is left. The AA are chewed off, and that again also happens in the process of integration. And so if you see a gene inserted into an organism's DNA and the target site is duplicated and there seem to be, well, the missing bases would be hard to tell unless you knew the precursor. But target site duplication is characteristic of retroviral integration. So we could look at genome sequences and know how many retroviral integrations there have been by just looking at the duplication. Because random integration of DNA, which happens, you know, the chromosomal DNA breaks and a piece of foreign DNA goes in, that doesn't duplicate the ends, but this does. So now the provirus is transcribed by the host, by host Pol2, and the left-hand LTR is the promoter for that, as you will see. The right-hand LTR is the terminator. And so the generation of LTRs is important because it generates a promoter for mRNA synthesis as well as a terminator. And then you get the genome, which is the complete mRNA. It's capped, it's polyadenylated, but of course it doesn't have an LTR at both ends. It's missing some sequence. It only has 2/3 of the LTR sequence, if you will. So let's see how integration works. The other enzyme in the particle. So we have reverse transcriptase, which with it is RNase H. They're both part of the same protein. And then a separate enzyme is integrase. It's a separate protein. And shown here is this beige blob. It actually is active as a tetramer. That's why it's written as IN4. 4 of the polypeptides are needed for this activity. And that protein catalyzes the integration of retroviral DNA into host DNA. How does it work? First, retroviral DNA comes in the nucleus from the cytoplasm with integrase bound to it. It's shown there on the left. Comes in the nucleus. It's going to interact with DNA via a variety of proteins, which you'll see in the next slide. The integrase has an active site in which there are 2 metals coordinated by appropriate amino acid residues. And the first thing that happens is the ends are chewed in a few bases. That's shown there. We're taking 2 bases off. It's called processing. And now we have staggered ends. We have a 5' overlap. The 3' end has been chewed back. And then the integrase associates with chromosomal DNA, which is shown in red. The integrase now has both DNAs, viral DNA and chromosomal DNA, nestling on it. The 3' ends of the viral DNA attack the chromosomal DNA and they nick it. That's how the integrase, of course, helps to do that. And that's also dependent on metal. So now you have nicked the chromosomal DNA and the 3' end of the viral DNAs have been ligated to the chromosomal DNA. And then now you would have on both strands a gap, of course. You would have a little mispairing between the viral and the host sequence, and then there's a gap. And the gap gets filled in by other host proteins, repair enzymes. So now you have the proviral DNA covalently linked to the chromosomal DNA. And in this process, you have a duplication of the ends because when you nick the host DNA in this upper right diagram, you now have both strands separated, so when you copy them, you're duplicating that sequence at either end. Now, integration can occur at many different places on chromosomal DNA, as you can see from this table on the lower left, but it's not random. There seem to be favored sites, and what is favored depends on the retrovirus. And here's an experiment where we're looking at percent integration. There are 3 different retroviruses here. There's a avian retrovirus, a mouse retrovirus, and HIV. And we're looking at the percent integration in different places, either within genes or within transcription start sites. And the first column is what you would expect if a DNA randomly integrated throughout DNA, chromosomal DNA. That's the number you would expect to hit a gene or to hit a transcription start site, 26 and 5%. You can see the numbers for the viruses are different. So the avian retrovirus, 42% within genes, so higher than you would expect from randomness, but basically random at transcription start sites, whereas the mouse retrovirus, very high for the transcription start site. So it seems to prefer integration at transcription start site. And for HIV, very high within genes and slightly above background within transcription start site. So that's what I mean, it's not random. It can occur, integration can occur in many, many places, but depending on the virus, they seem to prefer certain areas of the genome. So this means that in principle, the retroviral genome could integrate almost anywhere, and it could disrupt the gene. And we know that from gene therapy trials where we use retroviruses to deliver genes, that sometimes they disrupt the gene and may cause disease. And that can be a problem. So we have to modify the vectors to deal with that. Here's how we think these viral DNAs get targeted to chromatin. So on the left is our process of reverse transcription. We have the double-stranded DNA in the cytosol, and there's integrase bound to the DNA. So that's going to direct its integration into the host cell. And it's grabbing onto the ends, of course. There's a protein that enters the complex in the cytosol called BAF, which prevents the viral DNA from integrating into itself. Because when you think about it, this is a long DNA, and that the integrase could just have the viral DNA integrated into itself, which would be totally non-productive, of course. So this protein seems to prevent that. And then this complex goes in the nucleus. And here on the left, we're showing the integrase interacting with a cell protein called BRD that happens to bind nucleosomes. And so this is a murine retrovirus, which we think is targeted this way. So again, it's being targeted to chromosome by this protein-protein interaction, integrase with BRD. On the right is the way it works for HIV. It's a little more complicated. Here we have the viral DNA with the integrase tetramer, and this is interacting with a cell protein called LEG-F, which in itself will bind nucleosomal proteins, as you can see here. And so that will direct the integration to DNA wrapped around a nucleosome. But also HIV is further positioned by these other interactions. In this part of chromatin, we seem to have active transcription by Pol II. There's a promoter in red. Pol II is making a transcript. And this is CPSF6, which is a protein in the cell involved in polyadenylation. It's the cleavage factor that cleaves transcripts, and it's bound to the pol. And the viral integrase can interact with them to further position the integration at a very specific place. So the key here is that the integration reaction is directed to places in chromatin by integrase interactions with cell proteins. So we have viral DNA made in the cytosol, comes in the nucleus, it's integrated into the host DNA. The ends are duplicated, of host DNA are duplicated. We lose a couple of bases off the ends of the viral DNA. And now we have integrated or proviral DNA, which is transcribed from a promoter that is in the left-hand LTR. Now, both the LTRs are exactly the same. They both have a promoter for transcription and a terminator. Now, in the left-hand LTR, which would be there starting at the very end through the PBS, there's a promoter, but right next to it is the terminator. So that's not seen by Pol2. So Pol2 initiates and bypasses it and makes an mRNA, which of course we need to perpetuate the reproduction cycle. In the right-hand LTR, there is again a promoter and a terminator. This time, Pol2 recognizes the terminator and stops making mRNA. It's polyadenylated by the typical mechanisms, and now we have a complete message. That's how we specify initiation and termination. Remember, this is all done by host cell Pol2, recognizing signals on the retroviral DNA. On the right, there's a promoter and a terminator. I want you to keep that in mind, because if this provirus sits down next to a gene, it could turn it on by being transcribed. The polymerase might recognize the right-hand promoter and then transcribe into the neighboring gene. And you'll see that's one of the mechanisms by which these retroviruses cause cancers. We'll talk about that later. So I want to summarize this with a few key points. We start with two RNAs. We end up with one DNA produced by reverse transcription, which we think combines the best of both RNAs, if you will. There's a strong promoter in both LTRs built by reverse transcription. And the proviral DNA grabs onto the Pol2 transcriptional machinery to make viral mRNA, which is going to direct the reproductive cycle. And that RNA can be translated or can be encapsulated into new virus particles. So it's the full-length RNA has to get out of the nucleus. And remember, when we talked about splicing, I told you that this RNA can trick the cell into exporting it without it actually being spliced, because it has to be full-length, unspliced, in order to get into virus particles. What I think is really an interesting take-home from this is that really in this whole reproduction cycle, there's no viral DNA replication. There's no viral RNA replication. The only thing is reverse transcriptase, which makes one DNA from two RNAs. I wouldn't call that replication. It's reverse transcription. There's no RNA polymerase. There's no DNA polymerase. It's really remarkable. The cell is doing everything. Transcription is generating genomes by Pol2, and if the cell should survive infection and divides, both daughter cells will have the provirus. And as we'll see, that happens. These retroviruses don't always kill the cell, and the cells can keep on living and have that DNA in it forever, essentially. So let's summarize in the context of a reproduction cycle. We make DNA in the cytoplasm. The DNA goes into the nucleus, it's integrated. The provirus makes mRNA using Pol2 and promoters built into the LTRs. The full-length RNAs are exported and translated or packaged into virus particles, and the splicing gives rise to envelope glycoproteins. So if, as I said, the cell doesn't die, the provirus becomes a permanent part of your cell. Now, for HIV, this is a problem. Now, HIV typically infects CD4 positive lymphocytes, which we will see, it kills them. But in some cells, the replication cycle's aborted, so we're left with a copy of the DNA in long-lived lymphocyte precursors. And that's why you can never get rid of HIV once it's established this reservoir in you. We'll talk more about that later. There's no way to get this provirus out of a cell. People are working very hard for HIV, by the way, to figure out ways to get it out, but very difficult to do. The virus, the only way to get out is to make Pol2 transcript, and then it can make more virus particles, but the DNA is still left behind in the cell. And as we started sequencing genomes, we found in every genome sequenced, mammals, fish, everything, there are integrated proviruses. Genomes are littered with them. And if the virus happens to infect your germ cell, sperm or ova, then it is passed down from you to your offspring forever, essentially. So that's diagrammed on the bottom here. So we have a population of people, one is infected with a retrovirus. It doesn't have to be humans, it could be some other animal. One is infected with a retrovirus that goes into his or her germline. And if that person survives, then they have kids, and they spread the provirus to the offspring. So now you have many red offspring here. And you don't even know this is happening because you're not sick. The virus is, maybe you're not even making virus. As time goes on, mutations are introduced into this provirus. They may be inactivated, and that's the light pink, where they don't make virus anymore. They're just there, and they may be fixed. And some people may lose the provirus. And there are consequences of this, which I'll tell you about. But let's say if today some retrovirus were infecting the human germline, we would never know it until we sequenced the genome, and we would need to have tens of thousands of people inherit that provirus before we'd even detect it. And that would take thousands and thousands of years. So if HIV were endogenized, so this process of going in the germline is called endogenization. If HIV were doing that, we wouldn't be able to detect it for ages, but it's not because it doesn't infect germ cells. There are no receptors there. However, there are other interesting consequences of endogenization. So a retrovirus could infect your somatic cells, and once you die, that's the end of the infection. But if it infects your germ cells, that's a process we call endogenization. Then you will pass the provirus onto your offspring. And as I said, every genome sequence we've looked at is full of these endogenous retroviruses. And this is happening right now in koalas. Koalas, of course, are native to Australia. Whenever you see a koala in a zoo, it originated in Australia. They're not native to anywhere else. And they are being infected with a retrovirus which they got about 50,000 years ago from a rodent somewhere in Australia. A rodent retrovirus infected a koala. It spread. It then spread to other koalas. And now it's endogenizing the koala genome. It's going into the germ line, and koalas are passing these viruses to their offspring. So here on the right is a map of Australia. And the black circles are the percentage of koalas that are endogenized with this koala retrovirus, it's called. There are a couple of them, actually. You can see here in the north, all of the animals that were looked at are endogenized. The virus is already in their genome. But as you move south, not all of them are endogenized. So some have not either been infected or they're not offspring of the ones that are endogenized. With time, eventually, all of these animals will be endogenized. Of course, people are taking colonies of koalas that have not been infected and sequestering them. So we will have a koala retrovirus-free population. But we can see in real time the progress of endogenization, which, as I said, and people would take thousands and thousands of years. But we can see they have offspring. We can check the offspring and see that they've acquired these koala retrovirus proviruses. These endogenous retroviruses. So when I say endogenous retroviruses or ERV, ERV, I mean a provirus integrated into the germline. This is part of a larger collection of elements called retroelements in genomes. A retroelement is simply a sequence that moves around the genome via reverse transcription. You may know that there are various transposable elements that move around the genome. They're DNA that are copied and pasted somewhere else. Retroelements are a specific subclass of transposons that move via reverse transcriptase. And as I've told you, the proviral DNA in the germline is called endogenous retroviruses. We have a bunch of these. We have lots. They're all defective. None of them in us make infectious virus. They've been in us for so long, before we were even Homo sapiens, that they've mutated beyond making infectious virus. Beyond the endogenous retroviruses, 40% of our genome comprises mobile elements that move around by reverse transcription, retroelements. How does this work? So we have the gene in the DNA. It could be an endogenous retrovirus or not, something related. It's transcribed into mRNA, reverse transcribed, and then goes back into the genome via integration somewhere else. So that's a kind of a transposon, but it's a retrotransposon because it uses reverse transcriptase. And here's a table that shows you the extent to which the human genome is full of these elements. So again, retroelement is any sequence in our genome that moves around by reverse transcription. We have 42%, 2.7 million copies of retroelements. 42%. It's amazing. And sometimes these cause disease. Right now in the human genome, these don't seem to be active. Only less than 0.05% of these retroelements are active, which means that right now as I'm standing here, it's moving around my genome. But when they do, they can pop into a gene and disrupt it and cause disease. There are a number of human diseases associated with retroelements. Okay, so we have two kinds of retroelements, non-LTR and LTR containing. The name is quite straightforward. An endogenous retrovirus, so on the left are all the integrated DNAs to show you the different kinds. Endogenous retrovirus, of course, got there by infection. A retrovirus infected the cell. You got the whole viral genome there with LTRs and the GAG-POL-OM genes. So in humans, those are about 7.7% of our DNA is endogenous retrovirus. These are remnants of infections many, many thousands of years ago. Then we have what are called retrotransposons. These look very much like an ERV, but they're not ERVs. They lack the envelope. These are probably precursors of retroviruses. They were probably there long before retroviruses, and somehow the acquisition of an envelope allowed this to become a virus, because otherwise it's just a bag of membrane with protein in it, and it couldn't move from cell to cell. So we have a good number of retrotransposons in our genome. And then as we move down, there are other kinds of retroelements, lines, long interspersed nuclear elements, and that's a non-LTR, doesn't have LTRs. You can see different classes, almost 20% of our genome, line elements. These encode reverse transcriptase. They don't have LTRs, but they have a duplication at either end, which shows that they've been inserted via integration. And again, this, you make an mRNA, the RT makes a DNA copy, and then it will integrate somewhere else. So these move around the genome extensively. Then we have at the bottom here two kinds of elements, signs and processed pseudogenes. You can see the percentages there. They don't encode RT. They're moved around by RTs of lines or retrotransposons. These are mRNAs that got copied into DNA by another RT, and then they went back in the genome. And again, we know this because, look, there is a duplication of host DNA on either side. There's no RT, there are no LTRs. These are just mRNAs that randomly got copied by the reverse transcriptase that is in our nucleus. We have it in our nucleus, very, very low levels. All right, so those are retroelements. I want to give you a couple of examples of how these genes from retroviruses have been utilized by their hosts. One is called syncytin. And I say this has been exapted. When a host steals a gene from a virus, we call that exaptation. It's now used for something else. So as you know, the retroviral genome encodes GAG-POL envelope. The envelope is the glycoprotein in the particle that mediates fusion of the particle with the cell surface. So it's got a fusion protein in it. And that's shown in the left panel. Retroviral infection, the envelope binds a receptor. The cell and virus fuse. Okay, we know that so far. If you just put an envelope on the cell surface, that cell can fuse with another cell that has a receptor for it, independent of any virus particle. So this is what's happened with syncytins. Syncytins are proteins found in the placenta of mammals that have placentas.
Here's a placenta close-up. And, it's a place where you separate the maternal and fetal circulation. And part of the structure of the placenta is a syncytiotrophoblasts, this red layer of cells that is a key barrier between maternal and fetal circulation. This is a single fused layer of cells, hundreds of cells that have all fused together by producing this syncytin protein. And we've shown that in mice, if you get rid of syncytin, they can't make placentas. So many mammals have syncytins. They were derived by retroviral infections. So we didn't have placentas before retroviruses infected us and gave us this syncytin. It was originally an envelope, but now it's a syncytin, to allow the fusion of the cells to form a placenta. And this has happened multiple times in the evolution of primates, as you can see in panel A. So between 40 and 60 million years ago, there was one infection with a human endogenous retrovirus called FRD, and that gave rise to one syncytin, and that's present in all of these primates. And then there was a subsequent integration about 30 million years ago. Next time you think about a placenta, it wouldn't be there without a retrovirus. We'd be laying eggs or something else, maybe what the kangaroos do, right? They don't have placentas. But we wouldn't have placenta. We wouldn't have live birth if it were not for viruses. This is one reason I love viruses, right? It's amazing. So that's one example. Another example is HERV-K, which is a human endogenous retrovirus. You can see it in our genome. It's dead, it's not infectious, it's full of mutations, but it seems to be working. This virus infected our ancestors 200,000 years ago. There's no infectious virus produced, but during human embryogenesis, the viral mRNA is produced. It's produced from the 8-cell stage to the epiblast stage. So here's development here, right, up through 8, morula, blastocyst, there's the So this endogenous retroviral RNA is expressed precisely during this phase of development, which suggests it must have some role. You can actually see virus-like particles in these cells if you look at by EM, and this mRNA induces an antiviral protein called IFIT-M1, which blocks virus infection. We think it may protect the early embryo from infection. So another maybe remarkable activity of an exapted gene. There are plenty of other examples of this, but it's amazing how many things have been taken from these endogenous retroviruses. And the last one I'll tell you, which is just because it's so cool. If you've ever seen blue eggs, chicken eggs, they're blue because there's a retrovirus there. When farmers have a chicken that lays blue eggs, they breed them together so that they get chickens consistently making blue eggs. And what they are doing is that one chicken that had a blue egg, it had an avian retrovirus. All chickens in the world, by the way, are infected with avian retroviruses. We'll talk about that later. Retrovirus inserted upstream of the blue pigment gene, which is normally regulated. But remember, that right-hand promoter of the LTR turns on the blue pigment gene and the eggs are blue. And that's why chicken eggs are blue, because a retrovirus has turned on the blue pigment gene. I think this is so cool. I wrote a blog post about this, and a chicken farmer called me and asked if you could get rid of these retroviruses. But of course the answer is no, because they're in the genome, but they make blue eggs. They don't hurt you at all. So without retroviruses, we'd be laying blue eggs. No, white eggs, right? Because they wouldn't be blue. All right, our next question is, which of the following statements about retroelements is not correct? There are many copies in eukaryotic genomes. They are currently entering the koala genome, germline. Those in the human genome produce infectious viruses. They can be beneficial. None of the above, which is not correct. All right, how do we do? Yeah, the one that's wrong is they're in the human genome are making infectious viruses. None of our endogenous retroviruses are producing infectious viruses. They're all defective. There are many copies in eukaryotic genomes. They are currently entering the koala genome. They can be beneficial. Syncytin, the embryonic protection, etc. By the way, I just got a notice on my phone that stocks are crashing because of the coronavirus, right? Do you think they're crashing because Wall Street cares about us and us getting sick? Of course not. They're crashing because it'll disrupt economic productivity. They don't give a damn about us, right? All right, so I want to talk about the other
family of viruses that have reverse transcriptase in their reproduction cycles, the hepatinoviruses. And these are interesting because they have DNA in the virus particle. So hepatinoviruses we've encountered, they are DNA-containing viruses. Their DNA is weird because it's gapped. It has a protein attached to one end. It has a piece of RNA attached to the other end. And today you're going to learn why the genome is so messed up, which I promised you on day 2. These are envelope viruses. There's an electron micrograph on the left. It's spherical. It's an envelope around a icosahedral nucleocapsid that contains the viral DNA. And if you're curious about what this is on the right, it's just a transcription map where it's showing all the different mRNAs that are made from this genome. But this protein that is stuck on the 5' end of the genome, that's reverse transcriptase. You're going to see how it gets there. So these viruses bind cell receptors. They fuse at the surface, and then the nucleocapsid comes in. And of course, that has DNA in it. It's gapped, though. The nucleocapsid binds the nuclear pore. The DNA goes into the nucleus where it's repaired. Protein's taken off, the RNA is taken off, and the gap is filled in, all by cell enzymes to end up with covalently closed circular DNA, which is then wrapped around histones. In the nucleus, so now it's able to be transcribed. Couldn't be transcribed before it was fixed. It's transcribed to give unspliced as well as spliced mRNAs, which encode a variety of things to make new proteins, new capsids, of course. There's an envelope glycoprotein in there as well. But what I want to focus on here is that we take one of these full-length mRNAs transcribed from the DNA. It's out in the cytosol. The capsid protein is made, and capsids are assembled, and they contain the RNA in them. So the RNA is what is initially packaged into the capsid, very much like retroviruses. But I describe these as impatient viruses. As soon as the capsid is made and the RNA is put in it, along with a molecule of reverse transcriptase, these particles begin reverse transcribing, and they make a DNA copy of the RNA before they even leave the cell. Now, the retroviruses package RNA, they're released from the cell as RNA, and they do not reverse transcribe until they infect a new cell. But for some reason, the hepatinoviruses start to reverse transcribe as soon as the particles are made in the originating cell right there. So that's what I mean. RT commences in the cytoplasm, and then what's released has DNA in it. But as you will see, the process can't be completed because once you make a closed shell, this is my theory. Once you make a closed shell, you can't get any more dNTPs into it. They cannot permeate the shell. So when you make the shell in the cytosol, it captures a certain quantity of dNTPs, and that's all you have to make a DNA copy. And so the RT runs out of dNTPs basically, and that's why it's incomplete. So let's see how that happens. There are some very similar aspects of this process to retroviral reverse transcriptase. There are template jumps, for example. So there on the upper left, that's the RNA, the full-length RNA that's packaged in the particle in the cytosol. And it's packaged along with a molecule of reverse transcriptase. And again, as this RNA is packaged in the cytosol, the RT becomes active, makes a little bit of DNA on that stem loop structure there shown in blue, and then the DNA hops to the top strand. You can see there, it's very reminiscent of reverse transcription. It makes the first strand of DNA, and of course, as we are making the DNA, the RNAse H activity is removing the RNA template, so this RT has RNAse H activity, and there's our first strand of DNA. What's left behind is a little piece of RNA, which is going to prime the second strand of DNA synthesis, very reminiscent of reverse transcription. So we've got the first strand made, and now if we go up here, what happens is this RNA primer jumps to the 5' end of the genome and begins to copy it. And then, of course, there has to be a template jump once the reverse transcriptase gets to the end there. And then the bottom, step 7, is basically where it ends up. We've got a complete first strand of DNA.
We've gotten rid of most of the RNA, except there's this little bit of RNA here on the second DNA strand. That was the primer that's left over. And we've made a little bit of second strand DNA, but as I said, the enzyme runs out of dNTPs because it's closed in a particle and I can't get any more. So we end up with this gapped DNA. It's all one strand, just a little bit of the other strand. The RNA is stuck on it, and there is reverse transcriptase also attached. So that is what ends up in the virus particle. And this, of course, all happens before the particles leave the cell. So that's why you have a weird hep B genome, because it starts reverse transcription before the particles leave the cell. It doesn't have enough dNTPs to complete the process. In contrast, retroviruses wait to get into the next cell to begin reverse transcription, at which point the nucleocapsid has been disrupted, as I said, enough to let dNTPs in. But that can't happen here because it's very early on in the producer cell. All right, so I want to summarize all of this by showing you some comparisons among different kinds of viruses that have RT in their reproduction cycles. So today we have talked about retroviruses, and most retroviruses package RNA. And the yellow is what is in the virus particle. So that's RNA with a tRNA primer. And these viruses get in the cell, they make a DNA copy, which integrates and leads to the production of more RNAs, but those are packaged. And then I showed you the hepatinoviruses, where they package initially the genome RNA, but it gets reverse transcribed in the particle, so that what ends up in the particle is a gapped DNA. So retroviruses are RNA viruses. Hepatinos are DNA viruses because of when they started reverse transcription. Now there's a subset of retroviruses called foamy retroviruses, where a good fraction of the particles have DNA in them. Not all of them, but some of them do. Again, these start out by packaging RNA. Some of them reverse transcribe before they leave the cell, just like the hepatinoviruses do. And then finally, the cauliflower mosaic virus has reverse transcription in its reproductive cycle. It packages DNA. What's in the particle is DNA, but initially it packages RNA, just like hepatinoviruses. And before these viruses leave the cell, they reverse transcribe it. So what you find in the virus particle is DNA. So depending on where reverse transcription occurs can determine whether you are a DNA-containing virus or an RNA-containing virus. We now reach the end of molecular biology of this course. We're going to talk about assembly, making new virus particles next time, and then shortly move on to disease.